Hi folks, just a quick refresher on blood and tissue typing associated with the lab we did this week. So remember from what you know about the immune system that our immune system, part of our immune system's job is to patrol the body for foreign antigens. Remember, antigens are cell surface molecules. Right? Self-antigens are how your body recognizes its own cells. Foreign antigens are how you recognize pathogenic or just simply foreign antigens. Incompatible tissues are tissues that are recognized as non-self. And when you combine when you have donation of incompatible tissues to a recipient, you end up with tissue rejection, right? And in the case of incompatible blood types being mixed together, you can end up with an immune response that can be life-threatening because of the agglutination reaction. So there are about 35 different blood groupings, in addition to the ABO and RH blood groups that most of us are familiar with. But the frequency in human population is so low that we don't routinely type blood for those. Each blood grouping is based on different cell surface molecules. So this image is of part of a red blood cell. You can see the hemoglobin molecules in red, the membrane of the red blood cell, along with some of the cell surface molecules. And out here you have plasma, which contains a whole array of molecules, including antibodies. So antigens are cell surface molecules. Antigens can be proteins or polysaccharides. They're often a combination of both. And again, it's important to remember that the term is used in two ways. So if you're answering questions that have to do with antigens, you want to be sure to pay attention to whether or not what's being described are foreign antigens or self-antigens. And the term antigen actually is a contraction of antibody generating. So antibodies are proteins that are made in response to foreign antigens and not to self-antigens. And the binding between antigens and antibodies is dependent, like most things, on the molecular level on shape. Another basic term, you guys probably know this, but just to make sure, a blood transfusion is transfer of blood from one individual to another. Another basic term to have in your repertoire and vocabulary is agglutination. Focus on the sort of the glue part of agglutination. It's the clumping reaction that you observe between antigen and antibody binding. So again, antibodies are Y-shaped structures that have two separate binding pockets. So if this cell has an antigen that fits, and this one does as well, right? That's the beginning of a clump. Add lots of cells and lots of antibodies, and you can see why you might actually get clumping that's visible to the naked eye. So this agglutination is both the reason why mixing incompatible blood can result in serious trouble for the blood recipient, and also is used in the context of determining blood type, right? So if that agglutination is such that it becomes, there's so much of it that it becomes visible, we can use it to determine someone's blood type, which is what we simulated in lab this week. So, blood type. What does it mean? Blood types are named. We should say they are defined by the antigens, the self-antigens that are present on the surface of the red blood cell. Now, one of the things that can be kind of confusing about blood typing is that we have antibodies, we make antibodies to antigens that we don't have. Right, so as an example of that, if someone has type A blood, so here's the red blood cell, and that's the A antigen. Let me get a little triangle because it's kind of like an A. For reasons that people did not understand for a really long time, this person is going to make antibodies that will fit the B blood antigen. Right, so when you're trying to figure out sort of what's what here, it's critical to remember that blood type A, B, or O is determined by the antigens on our own cells, and that we make antibodies to any antigen, either A or B, that we don't possess. That's crazy on the face of it. If you think about the immune system, it shouldn't be happening, right? And the reason why, we now understand the reason why it does is because there is a bacterium that has antigens. Those are supposed to be little bees that are very similar in shape to the human B blood antigen. Similar enough that if we're exposed to these bacteria, right, we make antibodies to them. So when we say there, somebody, if I have type A blood, I make B antibodies. Well, you're not really making antibodies against the human B antigen. You're making antibodies against this bacterial antigen. And you might think, all right, Cindy, you said other bacterial cells have the A antigen. They do. So how come if I have type A antigens, I don't attack my own blood cells? And the answer goes back to the adaptive immune system. The thymus educates the immune system about self versus non-self. So if you're exposed to this bacterium, your thymus is not going to allow the production of antibodies that could potentially lock onto it, right? It's not going to happen because that's going to be recognized as self. And that's what this is all about. Okay, so this is just sort of laying out for you in table form using diagrams as well as words, right? This is they're saying the A antigen looks like, so that's what the A red blood cell would look like. The antibodies are going to be made not against A. So anti-A means an antibody against the A antigen. But they will be made to the differently shaped B antigen. When you're trying to figure out what blood types are compatible, there are two things two sort of rules of thumb to remember that you should memorize just because that's very simple. People who have type AB blood, in terms of the ABO group, they're called universal recipients. That didn't work. Recipients. Sorry, this pen is not working. Which means they can get blood donated to them from A, from B, from AB, as well as from O. Right? They can get blood from anybody without a problem. Type O are universal donors, right? Because they don't have the extra sugar group on top. They just have the little stick-like part. So, universal donors. So, they can donate to themselves, they can donate to AB, they can donate to B and to A, right? Type A can donate to type A and to type AB. Type B can donate to type B and to AB, right? I would not try to memorize this, right? Because this gets more confusing. I think it's easier to have the basic concept in your head and work it out that way. Okay, so these are standard kinds of questions. If you have type O blood, can you accept type A blood without a problem, without agglutination? So think about it for a second. Do. Okay, the answer is no. Why? Because type O does not have either A or B, so they're going to have antibodies to both A and B, right? So if you give them type A blood, there's going to be an immune response, agglutination. And agglutination is going to cause lots of trouble. It's going to be the same as having clots. Why can people with AB blood accept more blood types than people with O, A, or B? Well, a very superficial answer is because they're universal recipients, but that's not really an explanation, right? The explanation is that they have both the A and the B antigen on the surface of their cell, so the thymus tells the immune system, do not make antibodies against the A antigen or the B antigen, because that's who you are. So even if you're exposed to the intestinal bacteria or influenza virus, whatever it is that is similar in shape to the human A and B blood antigens, you're not going to make them. That does mean, by the way, that if you have AB blood, you're more likely to suffer from infectious intestinal diseases. But hey, you're a universal recipient. So, next question, which blood type is able to be used most often as a donor blood type? Why? Well, O, because you don't have the second story on the antigen house. And what that means, and the second story is either the A or the B antigen. So you can give type O blood to anybody without stimulating an immune response. One important thing that I want you guys to just sort of put in your head with this is that for reasons that you'll eventually learn about, you're going to disregard the donor antibodies. So pay attention to recipient antibodies and donor antigens. And that's what will allow you to determine compatibility. All right, so what's up with, forgot about this slide. Hemo is blood, lytic, lyse means to break. These are systemic responses to an incompatible blood type. Depending on the amount of blood that's transfused, it can be quite serious.
The reason that we care about the Rh factor is that the Rh factor is genetic, just like the ABO factors are. If a woman is Rh negative, so she doesn't contain, her blood cells don't have that antigen, and she reproduces with someone who's Rh positive, the fetus may be Rh positive. Now, for that first pregnancy, in general, if it's an uncomplicated pregnancy, everything's fine. The problem comes after labor and delivery, because at that point, the blood of the mom and the baby, actually, birth is a bloody thing. So the two circulations mingle. At least the baby's blood often mingles with the mom's blood. So the mom then produces antibodies, right? She's Rh negative. She produces antibodies that will fit the Rh antigen. So if there's a second pregnancy, her immune system has already been sensitized to the Rh factor. And this particular kind of antibodies, they can cross the placenta and they'll attack the red blood cells of the fetus. And that can lead to miscarriage and a whole lot of hurt. So this is just showing you up close and personal. There's umbilical cord with the blood supply going to the fetus, and then the mom's blood supply. And normally what's sort of a mustard color here, the chorion separates the blood circulation of mom and fetus. Now, antibodies can move across that, and that's important, because that's part of what protects the fetus from, because the adaptive immune system doesn't really develop until after birth. It's what protects the fetus from things like influenza. It's why, for most vaccines, women are recommended to get them. During birth, you get the rupture of the chorion membrane, and then the mom's blood becomes sensitized. Then, in a second pregnancy, that fetus can be at risk. This is referred to as hemolytic, when this happens, hemolytic disease of the newborn, or erythroblastosis fetalis. And this is a blood smear from a baby that had this condition. And one of the huge things to notice is that you have all of these red blood cells in circulation that still have nuclei. Remember, red blood cells normally jettisons, they throw out the nucleus before they enter circulation. But the baby is essentially, the cells are starving for oxygen. So the baby's bone marrow ramps up production of red blood cells, with the consequence that you end up with a lot of immature red blood cells in circulation. This is why, if you are Rh negative and you know that your partner is, or the father is Rh positive, or perhaps you don't know, but in the case where the father's blood type isn't known, women are given a nice big shot of RhoGAM. And RhoGAM is essentially antibodies against the Rh factor. So you might think, why the heck would you do that? Well, first of all, you don't do it until labor and delivery. What happens is, if the fetus is Rh positive, my fabulous drawing of a red blood cell with Rh positive antigens. The antibodies essentially cover all of the antigens that are present on any fetal blood cells that are in circulation. And that essentially, it's like an invisibility cloak. It keeps the mom's immune system from ever actually interacting with the Rh antigen. And so the mom can have a second pregnancy without worrying that the second child would be affected. This is what's referred to as passive immunity. So, active immunity means that your body, your own immune system, is producing the antibodies. Right? Your B cells are producing antibodies. Passive immunity is when you are given immunity by in this case, by being given antibodies against Rh. And so passive immunity, right, your immune system is still unaware that it's ever seen the Rh factor, right? In this case, that's a good thing. But in other cases, for example, if you snake antivenom is usually antibodies to the venom, right? It wears off. Eventually, those antibodies are gone. It saves your life, right? So one thing you guys need to be able to do is interpret blood typing results. The way to read the blood typing is to compare the control, which is just a drop of blood, to a drop of blood that's been mixed with antisera to only a single antigen. If you see clumping, which we see here, when you add antisera to the A antigen, or antibodies to the A antigen is another way of saying it, then you know this person has the A antigen. You have a separate blood drop that you mix with antibodies against the B antigen. You see clumping, so they have both the A and the B. And then, right, if you compare this, the anti-D, D again is same as Rh, right? There's no clumping here, at least compared, right, in comparison. And this is why it's important to have a control drop of blood. And one reason why I don't use the blood typing kits that we have in lab, the simulated blood typing kits, because they don't have a control lane. And especially when you're first learning to do this, how do you know what it's supposed to look like? So you need the control. So this is a more complete explanation of um, of how to read them, although it doesn't have a control lane.